Thanks so much, Dr. Lin. Thank you for the invitation, and thank you everyone for your time and listening to my presentation. So we tested before. We believe that you are all seeing my screen and my pointer that I used to present to some portions. I'm going to present. It's a two-parter. I was asked to present or to answer how the Kidney Precision Medicine project can be used as a resource for your own research. So I will do my best to present what is available on the KPMP portal. And I also will present my main research, which is how we can use those molecular signatures on niches to identify endothelial sulfates in diabetic kidney disease. So my disclosures is NIA funding, and I'm also funded by the Dialysis Clinic Inc. So essentially about those two consortia, right? So you might be familiar, given this is a network for NIDDK, but there's the Kidney Precision Medicine Project, which is we call the KPMP, which the goal is to understand the drivers of acute kidney injury and chronic kidney disease. And for that, we have a plethora of techniques, methods to interrogate the tissue. We have transcriptomics techniques, protein, metabolomics. We have multiple imaging methods. We also look at the epigenetic and also clinical and the pathology, right? The histopathology of those specimens. All of those specimens are altruistically donated biopsy specimens. So those are patients that they had not clinical indication for a biopsy at that point in time, and they donate the biopsy for research. So it's a treasure. It's a very unique opportunity to be able to work with this tissue. So this is all about kidney disease, AKI, and CKD, right? Conversely, the HubMap, which is the Human Biomolecular Atlas program, it's all about building an atlas of the healthy human body. So instead of focus on the kidney, focus on multiple organs. So we have 21 organs registered, one of which is the kidney, where we obtain a lot of reference tissue, right? So healthy cells in the kidney that we can use to compare with the tissue in the KPMP. We also have some healthy donors in the KPMP currently, but those are in fewer number than the diseased samples. So about the KPMP Atlas, so there is this publication that we published. Now it's a year and a half ago it was published, where we essentially we leverage all these technologies that we have on the KPMP, which include, single cell spatial transcriptomics with molecular data imaging to identify and characterize all the cell types along the kidney. We are able to identify 100 cell types, 6 cell states associated with different degrees of injury, and we have a total of 122 samples. I will not get into this study. I will present this as a portion of this that I use as a reference for my endothelial glomerular capillary studies that I will go following. I was just presenting this in the context of what we can find in the Kidney Precision Medicine Project Atlas. This is a project in partnership between the KPMP and the HubMap, so both of them are part of this particular study. So then moving on to the KPMP Atlas, right? So the website that you can use to help your own research. So this is the main page of the KPMP. If you search KPMP online, you'll probably be directed to this website. You can understand more about the study and about everyone that's part of the project. But then you can go to the Atlas. If you go to the Atlas, you go to this page where there's the kidney tissue Atlas. So we have 350 CKD participants, 78 AKI, 29 that are patients with diabetic disease, but that have not developed diabetic kidney disease, and 125 reference samples. So you can see breakdown by technology. on this particular samples. And then there's three main portions: the Explorer, the repository, and the spatial viewer. So the repository, it's the first one that I want to show, is where you can essentially download all the data that's developed on KPMP. It's all available. Dr. Just spoke before about the making the data fair, right? So that's the goal of this portion. Everything is available. You can search by participant. You can have age, enrollment, category. So the enrollment diagnostics, the adjudicated diagnostics, adjudicated by pathology, and etc. There's clinical metadata, and you can also go on top. Where I can, oops, let me close. I think this app's on the top. No, I cannot. Experimental strategy, that I was looking for. So if you have a particular experiment that you are looking, in our case, for instance, spatial transcriptomics, which is what I will talk about more today, it's all on the bottom. So if you click on spatial transcriptomics, you can find all the spatial transcriptomics samples that we have. Most of them are open, but like the FASTQ files and the BEM files are controlled to prevent identification of patients. So all of this is open. You can download all of this data, single cell, single nuke, multi-omic with attack-seq, codex. All of this you can download and use on your own research to complement your own experiments. This is the Explorer. So this is allows for a more quick test. So you can, for instance, look for NPH S2 or NPH S1, for instance, that encodes nephrin. And then you can see what are the technologies where this was identified. So you can look, for instance, on the single nucleus data set. And then you can see in the UMAP. So here's the UMAP of the single nucleus. Here is loading the single cell, it's loading the expression. So you can see that is mostly expressed in podocytes, as you expect. Some of those degenerative or injured cell types also expressed to a smaller extent. And then there's like some statistics across cell types. So you can use this to investigate some particular genes across all the different technologies. This allows for a more quick exploration. And then we have the spatial viewer. So this is so you can explore more of the spatial technologies. So I'm going to go again for spatial transcriptomics, which is the one that we produce on our side, so I am more familiar with. So I click on spatial transcriptomics, and then there's this sample ID. So once you click on the sample IDs, it loads a quick viewer that allows you to search genes. You can see the histology. You can see where here's the zoom of the histology. So you can see the glomeruli, for instance, and then you can search again NPHS1 or NPHS2, which are expected to be identified on podocytes of the glomeruli. And that's how you can, again, quickly explore those spatial technologies to see if any gene that you identify or any protein, if any gene that you identify is spatially distributed in a particular kind of region on like close to injury or close to fibrosis, and also like, for instance, a protein in codex, and so on. So essentially, this is what you can find on the KNET Precision Medicine Project Atlas. And I will stop a little bit to see if there's any questions about this resource that you think might be useful. If you think it might be useful for you, that I can help you understand.
So if you have any question right now for this part, please feel free to type the question in the chat or just let me know and unmute yourself. All right, I don't see the question.
Yeah, again,
sometime. Yeah,
I think, there's one I believe there's one, right?
Yeah, so there is one question from Dr. Swabanda. Do you have clinical biochemical detail associated with this data?
I believe also there is clinical metadata associated with the individuals, right? So to each patient there is clinical metadata. I'm not sure I believe this, like albuminuria, for instance, those are the, this is what you would expect or describe as biochemical, right? So those are essentially, there's participant metadata that you can search, right? And there's clinical metadata that you can also use to subset a particular portion of the data set. And you can combine, right? You can look for spatial transcriptomics of patients with a particular level of EGFR and so on.
Is that answer your question for Dr. Das?
I'm not sure if I can ask a question directly.
Yes, you
I can
can.
hear you.
Yeah, so my question was, like, for example, if you want to know in certain cell types, certain gene expression, one of your candidate gene expression is changed, and that is associated with changes in glomerular filtration rate or changes in the creatinine. So how you use this browser, to find out such information? Because I'm not sure if all of these are connected.
You're absolutely right. So you can, to some extent, do a very basic thing where you can break in this the Atlas Explorer. You can break down by basic AKI, CKD, or the resistors, right? And healthy reference. So that gives you some degree of if you can kind of see one difference. But and also here I think you have no, here you don't have any condition. So this is designed to a quick analysis, so you can have a preliminary feel. You cannot run statistics and look for differential expression. But if you go on the repository and you download the data sets, then you can absolutely control which data sets you are downloading and what is the clinical metadata associated. But then it requires a little more involvement to have to address this question specifically. Great question.
Is that answer your question, Dr. Das, or you have more question like based on your use case?
I'm fine, I think, at this point.
Okay. If you got more question, you can also ask at the end of the question session. So are there any more question about this part? So I think that's good.
Okay.
Yeah.
Yeah, I think I'll move forward. So I will start now talking about the research on the endothelial sulfates diabetic kidney disease. So we are on their network for NIDDK. So we all know about the kidney, that all the parts of the segments of the kidneys. We know that this is an example of the glomeruli. And one of the challenges that we have for spatial transcriptomics, particularly that I'm going to show later, is that we have all those cell types in close proximity. So we have podocytes, endothelial cells, and mesangial cells in the glomerulus. And we also have tubules and thick ascending limbs and macula densa surrounding all this structure, right? So it becomes challenging to identify exactly what we are talking about. So that's one of the challenges that we're trying to address. About the epidemiology of diabetic kidney disease. This is one study of 2,700 individuals in the state of Indiana from 2002 to 2015. 8.8% had diabetic kidney disease. And then the presence of some histological features are associated with progression to end-stage renal disease. And this will come again. I will look back to this at the end of my presentation. And then those are the two main pathological manifestations that I am going to talk about. One is the nodular mesangial glomerulosclerosis, where the mesangial cells expand and die, and we have this formation of these fibrotic nodules. And also the process of neovascularization, where we have an increase of neovascles that come from the efferent and efferent arteriole. That's also common in diabetic kidney disease. And it's not super frequent that's annotated histologically, right? Not always we have a report that comments on the presence or absence of neovascularization. So essentially, the study that I'm going to present start first with a case report, right? Where we were looking at the signatures of glomerular vascularization in a patient of diabetic kidney disease. This is a case report by the KPMP, so it's a KPMP project. And it is a patient, so it's 66-year-old female with diabetes, type 1 diabetes, and it was identified this had proliferative retinopathy, right? And you can see that there are those new vessels forming in the retina, and the patient had received VEGF induction to treat this macular edema. And in this particular patient, in the biopsy of this particular patient, we are able to identify histologically the presence of those neovessels, right? So you can see here in the surrounding the glomeruli based on the FFP or PAS. I think this is PAS. Then we use one of the technologies that we have on the KPMP, which is this three-dimensional tissue cytometry, right? So you can see here the staining for, or the immunofluorescence for CD31, which is a marker of endothelial cells. So here you have the diabetic glomeruli, and here you have for reference glomeruli, right? So for diabetic and for reference. And you can see that's not only an increase on intensity that you could, in theory, argue that might be a batch effect, but also the presence of those endothelial cells sprouting from the Bowman's capsule, right? And surrounding the glomeruli in this network of endothelial cells, of capillaries. Here is an analysis done with primaries where you can identify connected structures. So you can see that's essentially a single network of capillaries that is surrounding and penetrating this glomerulus. Conversely, in the reference, you have the efferent and efferent arterial, and that's what you get. That's what you expect that you have on a healthy patient. And then we also took spatial transcriptomics on this particular sample, and we compared the same four glomeruli that were analyzed before, and comparing with reference samples, we identified an increase in expression on multiple genes associated with neovascularization in this particular individual. So then we use this as starting point for our follow-up study, where we want to look for the regulation and transition of endothelial and mesangial cells, starting from a point of the spatial localization and spatial relationship of those cell types. This is a study that's currently under review, and you can find it on bioRxiv. So this is our questions, right? What is the role of those endothelial cells in the context of mesangial nodule formation? And are endothelial cells and mesangial cells changes in this context adaptive or maladaptive, right? So you have essentially growth factors and growth inhibitors of endothelial, epithelial, endothelial, and mesangial cells, and we want to understand this balance. And then our hypothesis is that the communication of those cells in the glomeruli will underlie the change in endothelial cells, vascular smooth muscle cells, and mesangial associated with not neovascularization and mesangial sclerosis. So to address that, we start with, again, going back to the reference that I talked before, the KPMP HubMap reference. We took the single nuke atlas and we essentially subsetted all the cell types that we expect to find, or the major cell types that we expect to find in the glomeruli. So we have podocytes, we have endothelial cells. The glomerular capillaries will be embedded here in this huge cluster. And also we have vascular smooth muscle cells and pericytes. The mesangial cells are also classified as subclassification of this particular cluster. So I took all those major clusters, and then we took the spatial transcriptomics and mapped that reference over here. So spatial transcriptomics, it's at this point already a more known technology, but essentially is spots. The tissue is the slide is covered with spots with 55 microns in diameter, and then the tissue is permeabilized, and the RNA is captured on those spots. And once we have the histology, we can identify what is a histological associated RNA signature. And then we use a Serrat-Lamb transfer method to identify what is the composition of the RNA composition of those spots. Because you can see each spot covers multiple cells. As I mentioned before, is a very mosaic-like structure, and we have to try to identify what is found there. And here you can already see that we have an increase in endothelial cell signature and a decrease in podocyte signature, right? This is essentially a forest plot looking at the odds ratio of identifying the signature in each of the condition. This is expected, right? There's podocyte effacement and loss of podocytes in diabetic kidney disease. And it also expected an increase in mesangial cells or vascular cells and endothelial cells, glomerular capillaries, due to the neovascularization. So here is an example that we use xenium to essentially on a diabetic sample to essentially identify what on a single cell resolution to identify what are the cells are the endothelial, the mesangial, and the podocytes, right? And then we are also able to do on a single cell a resolution to identify, again, this loss of podocytes, this increase on those vascular cells that express mesangial cell markers and the endothelial cells, right? So we subsetted only glomeruli and analyzed the cell types proportions. Here we have a much higher, it's much more, we have a much smaller confidence interval because we have way more cells. Conversely, on the spatial transcriptomics is in the level of spots. So then we took those cell types and we broke down into more specific cell types, right? So we have those vascular cells. We took those mesangial cells, those degenerative vascular cells, which are cells that are injured and lose some specific markers, and these non-specific vascular cells, so everything else that is on the atlas that we would not expect to find on a glomerulus, right? And on the endothelial cells, again, glomerular capillaries that we expect on a glomeruli, on the glome. The non-specific endothelial cells, which are all the other ones that we do not expect to find that are present on the atlas, and those degenerative endothelial cells, which are again cells that are injured. And you can see here that they kind of lose a lot of the specific glomerular capillary and the glomerular capillary markers and present some injury cell markers, right? So, but we don't know if those degenerative cells come from this NS bucket or the glomerular capillary bucket. So that's what we are including. And then when we look at the mapping of those cell types over conditions, we found before an increase on endothelial cells, but the glomerular capillaries do not increase to explain that. The cell type that increase are those non-specific endothelial cells, right? So we are trying to now understand what are the endothelial cells that have a signature, an unknown signature that map to the glomeruli, and we hypothesize that those will be the ones that are associated with neovascularization. So then we essentially use a neighborhood analysis or a niche analysis, right? So if you have single cells, you can understand the neighborhood or a niche as all the cells that surround a particular cell on a specific radius. You could do first contacts, but you could also define a radius and count all the cells that are there. On the spatial transcriptomics, we cannot count the cells because it's a single spot, but again, we are able to do that cell type mapping and identify those proportions. So we are going to use those proportions at what are the components of our neighborhoods. And then once we have that, we take all the spots that we have on the single nuke, on the spatial transcriptomics, and we cluster them. And we identify which clusters are each cluster of spots are particular niches. Okay, so you can see that some niches have more pre-glomerular cell types, right? They are more in the edge. Some might have proximal tubules or thick ascending limbs. Some have more fibrosis. Some have more podocytes. But I'm going to draw your attention to three particular ones. One that is more associated with diabetes, that is rich for endothelial cells. Those are all the three endothelial cell types: the non-specific, the degenerative, and the glomerular capillaries, right? And there are two neighborhoods that are associated with reference that are also rich for the endothelial cell types. But here we have a much larger composition of glomerular capillaries and much less of those non-specific cell types. So you can see the genes that we identify on each of one differential gene expression that we find on each of those. We have some genes associated with immune cells or fibrosis, right? And then what we did was essentially do a reverse mapping now, where we essentially, I will go to this slide first and then come back. We essentially took those neighborhoods and we mapped on top of the multiome and the single nucleus RNA-seq. And we have those three neighborhoods, the 1 and 2 endothelial reference neighborhoods and the endothelial DKD neighborhood. And in each of those two disaggregated technologies, we find a specific subcluster that is rich only for the DKD neighborhood, right? So that's where we identify which particular subcluster or subgroup of cells represent those proliferative endothelial cell types that you can find here, right? And you can see that those are the genes that are highly expressed on the proliferative endothelial cell types. And there are things like Notch genes, SEMA6A genes associated with this new cell type. So then again, the next thing is our hypothesis that cell-cell communication will be an important driver on these changes, right? So we went to investigate using the single nucleus dataset that has cell type resolution, what are the differences in the communication of those cell types. So here we have essentially communications with that endothelial cells are part of, right? Focus on some endothelial cells. And then you see that essentially communication between podocytes and endothelial cells preserve VEGF communication. It's not gone in DKD. And it's kind of, there's a change, but it's not, you have to balance the fact that there's a loss of podocytes there. Conversely, but actually not conversely, but we identified this particular receptor ligand pair here, SEMA6A, PLX, and A2, that is present on the reference between glomerular capillaries and injury endothelial cells, and also between endothelial cells and mesangial cells here, or glomerular capillaries and these non-specific cells. They're simply gone, right? So in the left side, you have the communication on the reference, but then on the DKD, this is gone. So this is communication that is known to be, it's known to prevent, it's known to regulate axonogenesis and prevent proliferation. So we believe that this pro-proliferative endothelial VEGF is maintained while this anti-proliferative communication is lost, right? So you have some other pairs that are also, for instance, DLL notch, that are also known to balance between sprouting and proliferation of endothelial cells. So then we focus on these particular changes of SEMA6A and PLX and A2. And here we look again, here on the spatial symptomics, you see that VEGF is present on the glomerular spots in both conditions. And here we identified that this is a reference sample where you have, this is to show that we have that we can identify the cells expressing endothelial cells and mesangial cells expressing. So here we have endothelial cells co-expressing PLX and A2 as predicted before, and here you can find both endothelial cells and mesangial cells expressing SEMA6A, right? And then what we did is not using cell type annotation, but the entire glomeruli, look at the density of those proportion of those transcripts, and then you can see that relative to the transcript of endothelial cells, because you have to remember that the number of endothelial cells increase, there's a decrease of SEMA6A, and then also relative to the endothelial and mesangial markers, or there's a decrease on the expression of PLX and A2 in the condition of diabetes. Again, validating what we identified before with the single node. So our next step, because we have that multi-ohm data set, was to try to identify which transcription factors might be involved in the regulation of this transition, this loss of SEMA6A and PLX and E2, right? So we look at, this is a trajectory. So we build a trajectory from this glomerular capillary to this injured endothelial cells, reproducing the process of injury. And then we looked at, with this, we built a transcriptional factor regulation network, essentially tell us what are the transcription factors that are regulating this transition. And then in this network, we look at a measure of centrality to identify what are some of the transcription factors that are central and that also regulate SEMA6A and PLX right? Because we identified that this transition was important. So we have this small network here of MEF2A, MEF2C, and TRPS1 that co-regulate each other and regulate SEMA6A and PLX and E2. So here, essentially, you can see the ataxic that has the open chromatin, and you can see the peaks where are predicted to bind on those on SEMA6A, and you see that's like a closure of those peaks from glomerular capillaries to this proliferative endothelial cells. You can see like this first peak here is gone, this peak here decreases significantly. Same thing happens for MEF2C, where MEF2A and TRPS1 are predicted to bind, but not to the same extent. And then here is this PLX and A2, where you can also see there's changes in the accessibility of those particular peaks. Like this one diminishes, I think, yes. Some of this MEF2C kind of maintains. We also did a similar analysis for the glomerular capillaries to those proliferative endothelial cells, which was not as good as the trajectory that we had before, but we also identified that those transcription factors are very central to the trajectory. So then, with that trajectory, we are able to produce an in silico knockout of this network, right? So here's the expression of the original expression of those genes, MEF2A, MEF2C, TRPS1, and then SEMA6A, PLX, and E2, and FCD4, which is another gene that we identified before in the receptor ligand. And this is after the knockout, so you can see that those three have zero expression as is expected. And you can see that there's this loss of SEMA6A and PLX and E2 along the endothelial cells. And also, this is the trajectory from the glomerular capillaries to the injured endothelial cells, the first one that I showed. And you can see there's essentially, it speeds up the progression, right? So this is the new development, the perturbed development flow after the simulation of the KO, the in silico KO. And you can see that essentially the cells progress faster to this injured phenotype. And we have, again, a similar disruption of the trajectory where when we have the trajectory for the proliferative endothelial cell, but then here essentially we have an opposite, so we have cells that will kind of go in this direction, and here they kind of, a lot of them revert to this more intermediary glomerular capillary, right? So that's why we started to identify that this network might balance this process of injury and neovascularization in the side of the cell fate for the endothelial cells. So then the next step that we did is to try to circle back our findings to those histopathological findings that we had before. So essentially, we took FFP samples. So all that was done before was done with frozen frozen spatial transcriptomics on OCT that we don't have great histology. But then we map those neighborhoods into the FFPE, where we have a smaller sample size, but we have a much better histology. So here you have the 7-micron H&E. This is the exact section where the spatial transcriptomic was performed. And this is a sequential section where I performed PAS. And all the glomeruli on those samples were individually scored based on the histology by a pathologist. And then what you can see, so here you have an example of those new vascularizations, kind of like what I showed before. And what we identified is that this DKD endothelial cell neighborhood, the first neighborhood that we identified, it is associated with the presence of with glomeruli with presence of neovascularization. So it is more likely to be found on glomeruli with neovascularization, right? So this neighborhood is associated with neovascularization. Here's another example of segmental glomerulosclerosis, where just essentially to validate, we can see this neighborhood that had more fibrosis associated with DKD that maps onto glomeruli with segmental glomerulosclerosis. And then, so we are able to align those identified neighborhoods with the expected histology. The next thing that we looked at was the MAF2C activity. Because we have that network and the target genes, a network of target genes, we are able to evaluate the expected activity of MAF2C. And then we can see that the MAF2C activity is associated with several of those histopathological manifestations that I mentioned before, so neovascularization, mesenteric expansion, KW nodules, and segmental glomerulosclerosis. And so again, you can see that it is always increased, the activity always increased in diabetic glomeruli, and it's further upregulated in this nodular glomeruli, linking the association of this transition of endothelial cells to mesenteric nodules, possibly. And then here is essentially, we are able to look, because we have single cell resolution on the Xenium dataset. So here you have reference glomeruli and glomerulus, and you can see that you have the endothelial cells here with some injured endothelial cells, but no proliferative endothelial cells inside the glomeruli. Here in blue, you have these mesangial cells. And then here you have an abundance of podocytes, right? So here's all overlapped with histology. And then conversely, on this particular one, you have this nodule here that I already move ahead and show that there's a lot of fibrosis surrounding and in this particular region here. And then you have this in the endothelial cells, you have those proliferative endothelial cells essentially penetrating and sprouting towards this region of fibrosis, right? And again, a much smaller number of podocytes are identified in this particular sample as we expected since our previous result. So finally, to essentially evaluate a possible clinical significance of that, we found a data set, or used a data set, single cell data set, where patients were treated with SGLT2 inhibitors. So then we took all the genes that are part of the MAF2C regulatory network, right? And you can see on that data set the change in expression between DKD and reference, and then between so we increase the expression of that network on most cases. On SGLT2 inhibitor, when compared to DKD, there's decrease of the expression. And when you look at the DKD patients treated with SGLT2 inhibitor, the direction of effect of this of the expression kind of returns to the same phenotype, or the direction of effect, at least, of the phenotype that we identified before. So in conclusion, we are able to identify this proliferative endothelial cell phenotype, so type, that's associated with neovascularization. We identify this anti-angiogenic communication that is lost communication between SEMA6A and PLX and A2. And we also find that MEF2C regulates those genes and also the transition between proliferative and injured endothelial cells, and the activity of MEF2C is associated with mesangial nodules. So these are my acknowledgements. So first of all, the three main labs, Eden, Ashker, and Dager lab that are part of the KPMP and the HubMap in our floor, and also the Indiana O'Brien Center, which is part of our group. Dr. Eden for the mentorship and guidance, Dr. Gish for the knockouts and simulations on expertise on Multiome, Dr. Cheng for the experimental side, using the spatial transcriptomics, and Dr. Phillips for the scoring of the individual glomeruli, and everyone else in our extended group in KPMP and HubMap that helped me along the way. And also, shameless plug, check our Indiana O'Brien Center and also the O'Brien Center Kidney Research Alliance, the OCRA, where a lot of those experiments can be provided to external users, essentially at cost. We don't do for profit. So essentially, if you have a kidney experiment that you think those things might help and other things might help, check. There's multiple kidney mouse models and multiple experiments that can be performed in this network. So with that, I thank you all for your time and attention and listening to me for this 45 minutes, I believe. And I will be glad to answer any questions you might have.
Yeah, thank you so much. Thank you, Ricardo. So now is our Q&A session. Everybody, if you have question, you are welcome to unmute yourself to join the discussion, or continue to use the chat function, and then we will repeat the question to let other people who watch the recording know about question, and then discuss at this time. Yeah. Okay, let me see if we receive some question during the presentation. All right, there is one from the audience. So one of the question from the audience, you mentioned that SGLT2 inhibitor in the summary that it may help restore the balance of MEF2C activity. Do you see this finding contributing to potential therapeutic targeting? I'm not familiar with the DKD, but just trying to understand, is this close to translating to treatment strategy?
So SGLT2 inhibitor is already a treatment that's used for diabetic kidney disease. So I think there's a more fundamental question of the role of endothelial cell proliferation on the context of. So we know that based on our results, those results point to the direction that the mesangial nodules are associated with this proliferation, right? But we don't know if the proliferation causes or tries to prevent the progression of those mesangial nodules, right? So I don't know exactly what we are trying. We believe that MEF2C can be a possible target to regulate the fate of endothelial cells, but we don't know yet in which direction we want to regulate, if that makes sense. And again, SGLT2 inhibitor is already a treatment used on diabetic kidney disease.
Okay, is that answer the question? Okay. Yeah. Are there any other questions from audience, or if you have your own research? Seems at the beginning, Dr. Ferreira introduced the resource from KPMP. So if you have any question about how to use this resource, feel free to join this discussion or ask question. Okay, there's.
So yeah, I can see it through in the chat. But go ahead. I was doing your part of the job. I'm sorry.
Yeah. Okay, so there's some comment, and then also there's question from Dr. Das. Can you predict causal mechanism that might be involved in SGLT2I mediated amelioration of MEF2C transcript network?
I cannot. I think I think causal mechanisms of SGLT2 inhibitors overall is an open question in this moment. But much less, I don't know I don't know how much of a direct versus indirect effect would happen in the network, right? So I don't know. I really don't know what could be the causal mechanism.
And then there is another question from Dr. Arturo. Are the data available for use in research?
Almost all of it, if not all of it. So most of this data was obtained from KPMP and PubMed. So both of them goes through a rigorous curation, right? So if any of the data that's used on this project is not, with one parenthesis that I'll get at, it's not available is because it's undergoing the internal curation process. It was all submitted to the main hubs already, but they might not be fully curated and available on those. It will be available on the same website that I showed before, right? The exception to this rule is the Xenium data. So the Xenium data is not part of the KPMP. So KPMP has a rigorous control of what are the technologies, because the biopsy sample is such a treasure. We have to have the technology approved first before we can apply to the biopsy. And those Xenium samples were not yet, so they are not on the portal, but it will absolutely be made available with the publication, the final publication of the manuscript. And if you have questions of the Xenium, feel free to reach out.
Does that answer your question, or you have more questions from Dr. Arturo?
It might be
I'm
a little
okay.
challenge. Oh, sorry.
Hello?
I'm okay.
Yeah,
I'm okay. I'm okay with the answers.
I was just going to say that it might be a little challenging to obtain exactly the same ones, because there's not in a single package. But on the bioarchive, we have all the samples, all the names of the samples and the participants. So you could go to that portal and search through the particular participants and obtain the exact same data that I used for the single nuke or the spatial transcriptomics and the multi-omics.
Okay. I think, oh, okay. There is one more question from the chat through the direct message. Now I see. So what he asked is, I joined the webinar a little late. Could you show me again how gene, protein, and metabolics are spatially distributed in different samples using your portal?
So I wish it was my portal. It's not. It's on the KPMP, so it's an NIDDK funded project. But essentially, you have so you can go to kpmp.org and then go to the atlas, so atlas.kpmp.org. So the repository, so there's these three big buttons. The repository, you can download individual samples. Going back to the previous question, you can download all the samples that I used in this manuscript. And then on the Explorer, you can look at single nucleus, regional transcriptomics, and regional proteomics to identify a particular gene of proteins of interest. And then there is the spatial viewer, let me go back here, where you can essentially select, for instance, spatial transcriptomics or spatial lipidomics or codex that is protein imaging. And once you click on a particular sample particular sample in this case of spatial transcriptomics, you can search genes or proteins, and you can link essentially histology with the gene expression or the protein expression or metabolites, for instance.